Good afternoon, ladies and gentlemen. Welcome to Carestream Molecular Imaging's presentation, In Vivo Molecular Imaging of RNAi Delivery and Efficacy. Due to the number of people here today, we have muted all attendees. If you have any questions during this program, please type them into the question box to your right. Where it says Send To, choose Send Questions to Staff. At the end of the presentation, we'll have a brief Q&A session. We'll try to answer as many questions as possible during this time, and we'll contact you directly if we're unable to answer any of your questions online. I am now pleased to introduce you to today's speaker, Dr. Seth Gammon, Global Product Manager for In Vivo Imaging Systems here at Carestream Molecular Imaging. Seth?
Thank you, Meryl, and welcome everyone. Today we're going to talk about RNAi and how imaging can accelerate RNAi research, in particular through looking at and studying the delivery of novel RNAi delivery vehicles and complexes to different sites in the body, including tumors, as well as using imaging as a way to evaluate how efficacious your siRNAs are likely to be at the target site, essentially using optical imaging for pharmacokinetics and pharmacodynamic studies. Just as a disclaimer, there are a lot of different patents out there that cover optical imaging, optical and X-ray, including patents by ourselves and others, and those you may need to be aware of these patents whenever you're doing your research. So let's get into what RNAi is. RNAi is the catalytic degradation of double-stranded RNA after short double-stranded RNA molecules have been processed inside the cell. Those short double-stranded RNAs get loaded into the RISC complex. They then target your messenger RNA of interest and then degrade that messenger RNA. As the protein is turned over inside the cell, there's no more RNA to make protein, and so your protein levels decrease over time. Now, this process occurs in nature as well as through use of synthetic siRNAs. RNAi was known in plants first through seeing these purple and white flowers. However, the mechanism of RNAi was really elucidated using nematodes, in particular C. elegans, and really rapidly Andrew Fire and Craig Mello got the Nobel Prize in Medicine in 2006 for their discovery of the mechanism of RNAi and elucidating that in nematodes. And they got this Nobel Prize very rapidly because the whole research community, particularly the biomedical research community, has recognized the importance of the ability to rapidly silence expression through exogenous delivery of siRNA in order to study basic biology without needing to make knockout animals. You can use this in mice, you can use it in human cell samples, it works in rats, frogs, zebrafish, fruit flies. And in fact, some of the more interesting and exotic uses are back in nematodes, where people are looking at using RNAi as well as a way to control nematode infestations in fields. There are three broad classes of RNAi, though this talk is really going to focus on siRNA. You can have large double-stranded RNAs which get taken up into the cells. These get processed by dicer, where they're chopped up into short double-stranded RNAis. They have a short-term effect at suppressing. The challenge with these is that they're very large, difficult to transfect, and difficult to make synthetically. On the other hand, siRNAs basically are chemically synthesized siRNAs that bypass dicer. Instead of chopping up a large double-stranded RNA molecule, they're already chopped up for you. These can be easily chemically synthesized or purchased from your favorite siRNA vendor. They can be modified chemically in order to make them more serum stable, for example, or prevent off-target binding. They still have to be transfected into the cell, although it tends to be a little easier to get these things in than large double-stranded RNA, and their effect is still short-term. Once they've been turned over, eventually their effect is gotten rid of from the cell. The nice thing about their ability to be chemically modified is not only can you label them with chemical constituents that increase their stability, you can label them with near-IR or visible fluorophores in order to track their distribution in vivo. Or if you want to look at these in humans eventually, you can label them with both fluorophores and chelates for radiometals. Another class of RNAi is siRNA, or short hairpin RNA. These are expressed from a vector. It could be a plasmid or viral vector. They are either transfected in the case of a plasmid or transduced into the cells. The nice thing about these vectors are that if you create a stably expressing cell line for these shRNAs, their effect at suppressing genes can be long-term rather than just short-term. Downside is that you still have to be able to select for those cells. And if you're trying to express an shRNA that actually kills the cells, that's a particularly difficult proposition. Beyond its use in basic research in order to study gene pathways by turning on and turning off various parts of a pathway and looking at downstream effects, RNAi has been proposed as a therapeutic for things like viral infections, high cholesterol, and cancers, because you can really, in theory, specifically target a certain subset of genes. Just like anything else, there are, of course, off-target effects. The endogenous microRNA pathway utilizes components of the RNAi pathway, and so those can become overwhelmed. And there's also significant challenges in delivery and proving efficacy of these RNAis. The delivery challenge lies around the fact that these aren't really small molecules. These are large molecules, and so they have a difficult time in perfused areas, and they have to survive in the serum. And then, of course, like any other drug, you need to be able to prove that it's efficacious, not only in vitro assays, but at the target site in vivo. Now, what I want to talk about today is how imaging can be used to accelerate RNAi research, and particularly when you get to the step of evaluating the efficacy and the distribution of either novel vehicles for RNAi delivery or novel RNAis or chemically modified siRNAs to target genes in a live animal. So one of the easiest ways, and one of the ways we'll look at today, is if you want to study the distribution, rapidly the distribution of your probe in vivo, see what are the major organs, target organs, how long does it take to clear out. It's really quite easy to now label an siRNA with a near-infrared fluorescent probe. If you're looking at a target vector, you could have it express a reporter. And if you want to look at efficacy or models of efficacy, you can have a reporter gene at your target site that you can track non-invasively in vivo, such as a fluorescent protein. Let's take a look first at how you can use near-infrared fluorophores in a really, quote, simple case of tracking just where do siRNAs themselves go, or at least chemically modified siRNAs. And in this case, the researchers labeled their siRNAs with Cy5, a near-infrared fluorophore. Working in the near-infrared is very important in vivo. There are other webinars where we talk a lot about this, but if you can, if you're working in vivo, work in the near-infrared. In this case, the Cy5 labeled siRNA was administered IV, and then its distribution in vivo was non-invasively monitored longitudinally over time with fluorescence and co-registered with X-ray in order to better understand where this fluorophore is going in the body. And as you can see, there's an initial broad uptake of the probe throughout the body with either labeled siRNA. And you can see that over time, it looks like it's being clear. It travels initially to the liver, as you can see here. Excuse me, as you can see here. Brief technical difficulties, I apologize. And then is excreted through the bladder. Now, what they noticed or hypothesized was that most of this probe was being cleared from the gallbladder into the large intestine and then out of the body as well. And so what they did was also do a bile duct ligation, as you can see in this animal and this animal, and you see an even more significant buildup in the liver over time at nearly all time points, which really backs up the hypothesis that one of the major routes of excretion for this is through the bile hepatobiliary tract. If you're doing optical imaging, it's always a good idea to confirm your results with an ex vivo analysis at some key time points, and the researchers, in fact, did that. You can lay out your organs on the platen of the MSFX Pro. There's plenty of room for multiple organs from multiple animals, and then observe where these go. And you can see that, in fact, whenever you do a bile duct ligation, that most of the probe builds up in the bile duct rather than traveling down into the intestines, as you see in the sham controls. As you notice from both of these experiments, there's very low levels of autofluorescence in the Cy5 channel, and in fact, if you move further out to Cy5.5 or Cy7, there'd be even less. And not only can you see it visually, but you can easily use the software tools to draw regions of interest around these different target sites and quantify the fluorescence light coming out and compare them. And they also wanted to go on and say, okay, well, we know basically how our siRNAs are being cleared. What happens if we modify or put these into larger nanoparticles? In one case, cholesterol nanoparticles, in another case, RGD, which is known to target neovasculature cholesterol nanoparticles. And in this case, what the cholesterol does is it increases your uptake into the liver and into the lungs. So by complexing your siRNAs with different nanoparticles or different targeting vehicles, you can change the whole body distribution of these probes. The other interesting thing here is that if you put in the RGD peptide, you can see that in the in vivo image, while you get much more liver uptake, just as you see with the non-RGD modified nanoparticles, in the ex vivo image you can see that the testes have taken up the siRNA labeled probe much more effectively than they had before. Other groups have also worked hard at looking at different complexes of siRNA in order to change not only the short-term uptake and distribution, as we saw in the previous slide, but in fact the long-term uptake and distribution. So here now we're not looking at just hours, but in fact days of uptake. So siRNA really doesn't have a lot of long-term uptake except in the submandibular glands and the pancreas. It's still being slowly excreted through the kidneys and to some extent through the intestinal tract. However, whenever you encapsulate these in polymeric complexes, you can really extend the bioavailability or the residence of these siRNAs in a lot of different organs. So if you need to, if your siRNA of interest takes a long time to take effect, for example, if your protein has a long half-life, you really need to make sure that you've got your siRNA delivered to that target tissue continuously, and these types of complexes may be of interest. This other group took it one step further. So rather than looking at just the distribution of siRNAs, they wanted to take their siRNA and label it for fluorescence to image it using both Cy5 and Di-647, but also put a targeting molecule directly onto the siRNA. So rather than bundling the target molecule on the nanoparticle, can I put my targeting moiety directly onto the siRNA itself? In this case, they're looking at fully conjugated siRNAs, and they wanted to see how robust is folate for targeting siRNA tumors. Now, folate has been shown to be able to target a wide variety of different tumors and deliver a lot of different payloads, everything from near-infrared fluorophores to radio tracers. And some people are experimenting with drugs, in fact. So it's not unreasonable reasonable to test out folate as a targeting agent for siRNAs as well. So here what you can see from the in vivo image quite quickly is a really nice little experiment. Here you have your Cy647 folate labeled siRNA. Targets to the tumor, and as you can see, it's being excreted through the kidneys here. However, whenever you look at a really nice negative control, where you have Cy647 siRNA, so everything else is the same but no folate, you really see very little uptake of this siRNA at the tumor site. And indeed, when you repeat the experiment with Cy5 folate siRNA, you still have really nice focal uptake of your probe at the tumor site in the mouse. Next, we want to move on from evaluating the targeting of the siRNA or shRNA to evaluating the delivery and efficacy of these siRNAs. So in this case, these researchers said, okay, like most people, you have to break the problem up at some point into two stages, the delivery and then the efficacy at the site. So beyond just being delivered to the surface of a tumor or a target organ, you have to get your siRNAs into the cell in order to be processed and degrade the messenger RNA where they have effect. So again, naked siRNAs without any delivery vehicle may not be really the right way to go. And in fact, this group was looking at polyarginine chitosan complexes with siRNA. And what they did is they took two tumors and they made sure these tumors expressed red fluorescent protein. And then they directly injected different complexes of siRNA, including naked siRNA, toward RFP or as a negative control to the PGL2 vector. So what you can see here is that with imaging, we can really rapidly evaluate how effective is my siRNA at targeting a mock target in vivo, a real protein that's expressed from mRNA, where the siRNA has to be delivered into the target site, it has to go inside the cells, and it has to effectively silence. And you can see that with their complexes, they were able to specifically target the RFP. If they put in a negative control, it does, such as SIGL2, it does not decrease RFP expression as observed by fluorescence intensity. And in fact, naked siRNA doesn't get into the cells as one might expect. Now, granted, this takes away the whole delivery problem. Another paper that's quite nice really wraps all of the principles we've talked about today together into one paper, where they were looking at a nanoparticle that's a multimodal nanoparticle. It can be imaged by MRI as well as by near-infrared fluorescence, as you can see from the Cy 5.5 labels. It has a targeting moiety, so we're not just going to all tissues. It's going to all tissues, but then hopefully being retained at the tumor site, as well as siRNAs to a specific protein, such in this case, GFP is my novel target. So what they do is transfect cells, stably transfect cells with GFP and RFP, implant one of each on the same mouse, and then systemically inject these nanoparticles, see if they can be targeted using the Cy 5.5, and then in a different emission filter set, look at whether the GFP can be specifically silenced in the presence of RFP. And so, in fact, you can get quite nice specific silencing and targeting. So, they were able to show with both whole body and microscopy that their probe was able to get to the tumor sites. And then you can look at, and this experiment quite elegantly designed, a very wide emission filter window that covers both GFP and RFP. So here, prior to injection, you see same tumor cell, parental cell line, 9Ls, same mouse, different size, GFP, RFP. 48 hours after injection, you see nice silencing of the GFP while, in fact, the RFP is increasing. Beautiful thing about fluorescence is you can then take out these samples, put them under confocal fluorescence microscopy, and see the same things. And of course, really the gold standard for measuring the efficacy of siRNA, whether you're talking about in vivo or ex vivo, is quantitative real-time reverse RT-PCR. And in fact, they were able to show that you have really nice selective silencing of the GFP. So in conclusion, I hope that you've seen that in vivo imaging really provides a versatile platform for answering some of the most critical questions in RNAi research in vivo. And that is, can I get my RNAi to my target site? And in fact, can I silence a target of interest in the presence of other targets? Using fluorescent proteins and other reporters as a model target works quite well. Of course, one could also create fusion proteins directly to your protein target of interest, so you have a imaging for a surrogate marker of degradation there, or as some part of a reporter that reports on a downstream process. Carestream's in vivo imaging systems, whether you're talking about the new MSFX Pro with high-speed X-ray and fixed lens, or our extremes, either the front-illuminated 16 megapixel system or our high-sensitivity back-illuminated 4 megapixel system, are really ideal for this type of non-invasive imaging of multiple mice at multiple time points. So I'd like to thank you all for your attention, and I would be glad to take any questions that you might have.